Yeah, thank you. So let me first thank the organizers for this wonderful meeting. It's great to be back at ITP or KITP. Actually, I've been here 32 years ago. Probably that was the first biophysics program at ITP at that time. It's not yet called KITP. There's a picture taken there. We didn't have this nice building. It was just a bunch of rooms in some other building shared with other departments. But that was a great meeting. It got a lot of people, a lot of physicists into the field of protein folding. Anyway, so okay, my group is interested in general in the past few years in understanding how the amino acid sequence of intrinsically disordered protein IDPs would affect the phase separation propensity and how they mix and they co-mix and so on. So there are many levels of theoretical modeling for this process at different level of structural and energetic complexity. So a lot of people use Florhagens or maybe overbeat one. These are mean field theories, so they're very useful, but they are basically dealing with composition. So you have two sequences of the same composition. You can't really distinguish them. So I'm going to talk a bit about this approach called random phase approximation, which can, in an approximate way, deal with the sequences. Well, we first apply it to electrostatic interaction, but I won't have time to go into it, but we can actually also generalize it to other contact interactions, including 20-letter amino acid sequence. And there are more advanced theories, so we're going to talk a bit about this too. One is field-theoretic simulation. The other is a generalized form of RPA. And of course, you can go all the way to experimental simulation using either coarse-grained model or atom model. So just a few words about this random phase approximation. So it's basically an approximation that you assume that the fluctuation in the density of the polymer is small enough so that it can be captured in Fourier space by a quadratic term. So we first applied it in 2016 to rationalize the very different phase separation propensity of this intrinsically disordered protein DDX4 studied by Julie Forman K's lab. Here's the wild type, here's the charge scramble variant. They have exactly the same composition, but very different propensity to phase separate. And our model can capture that in an analytical way. So we also will talk about ways to overcome some of the limitation of RPA, because RPA assumes a Gaussian chain approximation. So that's not always true, especially if you have a strongly charged polyelectrolyte. So we're going to generalize that as well. Oh, just a few historical notes. So the term random phase approximation is mysterious. So it first appeared in a study in 1951, a study of quantum electron gas, where this term was introduced. But of course, there are quite different kinds of meaning. Later on, like in this paper by Dugent, it was used to describe an approximation to deal with excluded volume. Okay, so in our case, we are using it in the sense that we're just using a quadratic term to describe the configuration, distribution, or multiple chain system, following mostly the work of Monica Oliver de la Cruz. The references are here. Okay, so I just mentioned that the Gaussian chain approximation may face some problem if you were a strongly charged heteropolymer. So we developed this approach called renormalized Gaussian, or RG. It depends on renormalized Kuhn length. This method was not first developed by us, it's by our collaborator, Kingsley Ghosh at the University of Denver. So he used a variation approach. So you first write down the full Hamiltonian with the electrostatic interaction, but then you assume that it can be written down in this form, just like a Gaussian chain without the electrostatic interaction, but with renormalized Kuhn length, and then you use a variational method to approximate determine this renormalized Kuhn length. So his Kuhn length is going from L to this renormalized Kuhn length, which depends on the sequence, and of course depends on the condition of the experiments and so on. So RGRPA is just a synthesis between this approach of renormalization Kuhn length and the conventional RPA approach. So basically, we replace Coulomb length with we normalize Coulomb length, which is a function of electrostatic interaction. This LB is the medium length and the sequence. So I'll tell you a bit about this application to understand the electrostatic interaction in the sort-dependent reentrant phase separation of the system Kaplan-1. So it has been known for some time that there are sort-dependent reentrance behaviors in IDP condensates. So here's an example with RNA. So only in the intermediate concentration of RNA, you get phase separation. And if you have high concentration or low concentration of RNA, you don't. And here is an impressive work done by some people in the room, looking at 6 proteins. And in these cases, if you have no sort, you don't have you have phase separation, and then you have intermediate sort, you don't have phase separation, but you have high sort, you again have phase separation. So why are we interested in this? Because this is a good way to probe the physics of phase separation, and perhaps it can also lead to some understanding of signaling. So ATP in assembly and disassembly of our molecular condensates, there are various roles that ATP plays. So this is an earlier study showing that ATP can dissolve condensates, but a later study showed that ATP can actually induce some kinds of reentrance phase separation only with the, for first C-terminal domain, only an intermediate concentration of ATP will give you phase separation. This is done by a group in Singapore. Okay, so this is a collaborative work with our Louis Kay and Julie Foreman Kay in our department at the University of Toronto. So it's looking at Kaplan-1, and we try to use our model to rationalize some of the experimental data that they obtained throughout the years. So in addition to Kaplan-1, we also look at the phosphorylated version of Kaplan-1, we call PY Kaplan-1. They are very different because Kaplan-1 is strongly charged with a net charge of plus 13, so it's a polyelectrolyte. And the phosphorylated PY Kaplan-1, if it's all 7 tyrosines are phosphorylated, it has a net charge of -1, so there's some difference. This distribution is phosphorylation, but basically it's more or less neutral. So it's a polyamphlite. So the RGRPA theory should be very well suited to do this because we can deal with polyelectrolytes and polyamphlite at the same time. So here are some of the experiments that Louis and Julie's got throughout the years. So Kaplan-1 doesn't phase separate without ATP and magnesium, but it's phase separate. But if you look at phosphorylated Kaplan-1, it's different. It phase separate without ATP, but it would dissolve if you add more ATP. And the other observation is ATPs are strongly co-localized within the condensate, as can be seen here. Okay, so here are the charge patterns for Kaplan-1 and PY Kaplan-1. So here's the RGRP prediction superlated with the experimental data. So the symbols are experimental data. So you can see that they have different sort dependence on sodium chloride. Kaplan-1 propensity, phase separation propensity, increases with sodium chloride, whereas PY Kaplan-1 decreases with sodium chloride. This is captured by theory. And ATP has a similar effect on Kaplan-1 and PY Kaplan-1. It has a reentrance behavior for Kaplan-1, but if you add ATP to PY Kaplan-1, it will just monotonically decrease the phase separation propensity. So the results that I should show you is a simplified version of RGRPA that assume that the salt concentration is the same outside in the dilute phase and also in the condensed phase. Here we do a more general calculation, so we get this two-dimensional phase diagram and timelines. And okay, here, let's first look at this. This experimental data showing that reentrance behavior for both ATP and sodium chloride. Sodium chloride doesn't go all the way down to dissolving all the Caprin-1 condensate, but the trend is there. Okay, you look at this phase diagram, you can see that RGRPA predictions are consistent with experiment. Here you don't have phase separation when you have 0 sodium, but then you go through a phase separation regime and then the condensate will dissolve again. And PY Caprin-1, you don't have this trend. And for ATP, you also have this trend. So since it's a theory, we are not sure that they really work. So we also try to verify this using coarse-grained molecular dynamic simulation. So you can see that it's working, at least for the general trend. So these are sort of a standard method using the KH potential for Chitan nitro, which is basically a Mierzauer-Jernigan matrix. And so if you increase the concentration of sodium chloride, you can see this critical temperature first increase and then decrease for Caprin-1. And if you look at a variant, which we also have experimental data for mutating all the arginine to lysine, you also get the same trend. And here you can see that sodium's a bit depleted within the condensate, which is expected because Caprin-1 is highly charged, positively charged, and chloride is attracted into it as a counter ion. Okay, so we can look more deeply into this. So here is just a snapshot showing that most likely that the way that this polyelectrolytes phase separate is because of this bridging interaction with the counter ion chloride. This is very similar to the effect discovered or illustrated more than 20 years ago by Panagiotopoulos's group using lattice simulation of polyelectrolytes on a lattice with counter ions. So we scanned through the snapshot, 4,000 snapshots and so on. We indeed see more than 87% of the chloride, the counter ions, are actually engaging in this kind of bridging interaction. So those are the things, those are the effect that is stabilizing the condensates for polyelectrolyte. Okay, so let's switch gear and look at another method that goes beyond RPA is field-theoretic simulation. So since we are at ITP, we can talk a bit about field theory. This thing actually has its origin in this seminal paper published in a Chinese journal in 1981 by Parisi and Wu, developed a method beyond path integral and a commutator method to quantize field theory. Basically, it used a Langevin equation in a fictitious time called the fifth time because space-time is four-dimensional. And so the quantum average would be the time average in this fictitious time evolution. So this method is later called stochastic quantization. So 2 years later, Parisi pointed out that this method actually can be generalized, but it has to be proved case by case to a Hamiltonian that's complex. And Crowder also made a similar observation. And since then, Grant Fredrickson, which is actually here at UCSB, did a lot of work to apply this method to polymer physics. And in fact, the first paper that applied this method to biomolecular condensate is done by Grant and John Scheer, who is in the audience, and in this paper in Journal of Physical Chemistry letter. Okay, so very briefly, how it works is that you start with a general path integral in terms of coordinates of the chains, and then you do a transformation to a field theory with a complex field dealing with the density of the beads of various chemical species. And then you use this Langevin formulation and turn it into a Langevin dynamics, and then you calculate the average in this time evolution in this fictitious time. So for those of you who are interested in applying this method, we do have some software that is publicly available, so you can go to this website and then you can download this and play around with it. Okay, so one thing that we are particularly interested in calculating in the field theoretical simulation is this pair correlation function of two polymer beads or polymer bead with some chemical species. And from the plot of this, you can see the trend of this re-entrant phase behavior for Kaplan-1. If this ATP starts from 10 to the minus 4 to 0.03 to 0.5, it goes something like this. It's not very clear, but you can see that it is actually going that way. But for the PY Kaplan-1, is monotonically decreasing if you increase the ATP concentration. And here you can see this is a correlation between the polymer beads and ATP. You can see there's a strong peak here showing that there's a co-localization of ATP with the condensate. So here's some field snapshot. Let's focus on this. So you can see that if you vary the ATP concentration, you first get some diffuse configuration and then a condensate and then diffuse again. So this is the re-entrance behavior for Kaplan-1. And then you can see that this is for ATP. The ATP is co-localized with the condensate. And this is consistent with experiment done by Lewis and Julie's group. The sodium is actually at a lower concentration inside. I forgot to mention, if you look at timelines, actually this is the trend that we got, although we can't get it to be that extreme, so there's something to be done with the theory too. For ATP, actually is strongly co-localized within the Kaplan condensate. Because we can calculate this pair correlation function, so we can actually use FTS, field-theoretic simulation, to calculate contact probability, contact maps, and so on. So contact can be defined by integrating the pair correlation function over a small volume that you can use to define a contact. And by doing that, we can rationalize this binding of ATP in a residue-specific way in Kaplan-1 condensate. So this is the experimental data from Lewis group. And this gray points are experimental data, and these red points are the FTS prediction. And they are basically giving the same trend. And you have more binding in the region that are more arginine rich. The other thing that we are able to do is to calculate the surface potential. The surface potential was measured by Lewis using some probe molecules. And so in the same spirit, we put in the system a very low concentration of test charge. And this test charge, you can calculate the pair correlation function that you can integrate the potential mean force, taking the log, and which is a potential mean force, integrate over that small volume, and then you can get basically how much ATP is bound to a certain residues along the chain. And then we also get the same trend as experimental data. Okay, so more recent applications. So because the rate of gyration of a chain can be calculated from internal distances. So FTS, because we can do the pair correlation function, it can be used to calculate rate of gyration, even for an isolated chain and certainly in a condensed phase. So we apply this. We don't have time to go into the details of this. And using FTS, RPA, and also cost-square molecular dynamics to try to evaluate the ability of these two popular sequence charge pattern parameter, copper from Papoose group and SED from Kingsford-Goss group. And we found that you look at RG, these are two different reduced temperature. SE is really very good. So this is over a bunch of sequences, similar to Rohit's set, but we also introduced some other sequences to challenge these parameters. But copper is not that good. This is because of some local, non-local effect that I won't have time to go into. But since both SED actually is designed for a single isolated chain, is actually not too surprising. It does very well, but for phase separation, their ability to predict the trend of the critical temperatures is about the same. As SED is slightly better, but not much better. Okay, so in the last few minutes, this is interesting, so I want to get into this. So this is a collaboration with Ben Sabari in UT Southwestern Medical Center. So this is looking at partition coefficients, see whether we can predict partition coefficient, in this case, into this MAD1 condensate. So we used RPA theory. So the outline of this is that we first assume that there's a scaffold of MAD1, and then we try to calculate this type parameter. And we first focus on electrostatic. So electrostatic has a zero order term, first order term, zero order term is just the net charge. And then first order term is this RPA expression that can take into account the sequence pattern. And you can see that from the known partition coefficient measured by Ben's group. If you just use the mean field theory, just net charges only, you get certain correlation, but it's not great. If you include this RPA, which takes into account the sequence pattern, it does significantly better. The R is up to 0.7. And so we use this kind of correlation to tune a few parameters in a model to try to predict partition coefficient into MAD1 for sequences that don't have experimental data. So this is a high throughput method. We can very quickly scan through more than 160,000, 200 residue windows for over this around 900 IDRs that Ben picks for their relevant to transcription. I don't know the experimental details, let me first confess. And so we pick out a field that's predicted to be high PC, and they are all confirmed by experiment. And a field predicted to be low PC, they're also confirmed by experiment. But there's a caveat. There's also a few cases that we found that we predicted to be low PC, but they still partition in a significant way. And in those cases, we found that they have a lot of aromatic in it. And so maybe the next step in generalizing this method is to include this other non-electrostatic interaction to make a more comprehensive, more general approach for this prediction. And for those cases, actually, when we add in this m pi pi potential function derived by Jerry, they are all consistent. Okay, just to recap. So the ATP effect on caprin-1 and py-caprin-1 is very similar to sodium chloride. So electrostatic is very important, although we should also look at other effects. The caprin-1 reentrance behavior largely coming from the short ions bridging effect, the counter ions bridging effect. And RGRPA can provide a uniform formulation for polyelectrolytes and polyampholyte. FPS can be used to compute sequence-dependent contact probability, rate of gyration in dilute and condensate bases, and surface electric potentials. SED is a better predictor for isolated chain than copper because it actually takes care of the local, non-local, the difference in local and non-local charge patterning for an isolated chain. But in the condensed phase, that local and non-local difference is less, so copper can do as well in the condensed phase. So this MAD1 case suggests that RPA can be used as a high throughput method for predicting large number of IDLs, the partition coefficients, into ideal scaffold condensate. Okay, thank you very much for your attention.
Yeah, thank you for this very interesting talk. So in the beginning, you said that you renormalize the Kuhn length along the chain.
Yeah.
Now, when you have an IDP, you have essentially a local Kuhn length, which varies along the backbone of the chain.
Right.
But in your theory, you probably have an averaged one.
Yes.
So by how much does it vary when you take a typical IDP? Do you have any idea?
I don't remember, but in general, you can derive a renormalized Kuhn length for different Kuhn length for different pairs. So in general, you have a LRIJ of a residue pair.
That's clear.
Yeah,
that's clear.
Yeah, so, but here for simplicity, we just do it with an average. I don't remember
because
how much.
Depending on how much it varies, that would give you some indication of the validity of your approach, wouldn't it?
Yeah, that would be. But we haven't tested it yet.
Thank you.
Really wonderful talk. So one question I have is about the salt partitioning. With the exception of one of the phase diagrams that you showed, it looked like almost all of the calculations and the simulation that you showed explicitly have overall salt partitioning into the condensed phase, or at least for the examples I caught. So to what extent does the theory predict that in general for this class of systems, and to what extent do you think that matches the data?
So the sodium ion is actually lower in the Kaplan.
Total salt. So the sum of the two microions.
Oh, the sum of the two.
Sum.
Okay,
Yeah.
I haven't looked into that. So
okay.
Yeah, but in general, it shouldn't be a general feature because the ions and the salts they are sort of treated independently. So that shouldn't be, but I don't know. I haven't noticed that. Thank you. Yeah.
Very elegant calculations. Wonderful work. I have a question regarding the bridging interactions. I was wondering whether the bridging you see is specific for arginines or whether lysines, for instance, also exhibit bridging.
Yeah, lysines would also exhibit it because actually in this model, we can't really distinguish them, right?
Okay.
Because it's all sort of just a charge, right? So I guess if you run a atomistic model like Dr. Zhao did, then maybe you can see the difference.
Yeah, because I was thinking we should look in our atomistic simulations whether we see the difference.
Yeah, because they say with arginine you may have other interaction that would bring them together already because of the pi interaction and so on. So that may be different.
I was thinking that might be influenced, but you don't capture that.
I didn't look into that.
Thanks.
I'm kind of curious whether this can be extended to like chromatin, so like in a way that can help us understand chromatin organization.
I haven't tried it, but it could be. But you may have some way to deal with the persistent length and things like that. Okay. Because this is still very much of a Gaussian light chain model.
Okay.
Last question.
That was very interesting. It's amazing that you could bring so much physics to the sequence dependence. Is the RPA approximation good enough to get counter-ion release when oppositely charged residues come in contact, like in Ben Schuler's talk yesterday? I know in simple systems that's a way of gaining a few kT of energy. Right. When a plus and minus come together, the screening counter-ions get released. And it's a little bit of not a direct quadratic effect, but maybe when you put in your effective Hamiltonian and you get your quadratic, you can get it. But do you know if you get counter-ion release using that approximation?
I think so. I mean, we don't have ion condensation, but we do have different partitioning of different charged ions into the condensate. So in a way, the ion release is there.
But it would mean that you have to see then that the plus minus residues are in close contact. It's not the question of bridging by an ion, but the question of plus and minus. Anyway, that might be interesting because counter-ion release theoretically is a little bit subtle. Right. It's not a direct interaction.
Thanks, speakers again. So the next talk is